key element? Carbon. And typically in biology we say carbon and hydrogen both. But the key element is carbon. So a simple carbon chain consisting of carbon and hydrogen, that is the simplest of the organic compounds. But there are different groups that can be added to these carbon chains that would then give them specific functions, and that's why these are called functional groups. These are not all of the functional groups. If you take organic chemistry in college, you'll learn other ones. But all of these are going to come up when we go through carbohydrates and lipids and proteins and when we talk about DNA. So that's why we're going through these specific ones now. So you will not, on the AP exam, at least as far as I've ever seen, have to draw these from scratch. All you would need to be able to do is to match a drawing to the name. So you should be able to match the name to the drawing. And the other thing is, and I may have told you guys this yesterday, that's sort of like how they use x in algebra as a variable. In organic chemistry, a lot of times we use r as like a variable. So, for example, the hydroxyl group. So the hydroxyl group, I'm going to draw R, which is my little variable, is OH. That is the formula for hydroxyl. And the R would represent whatever it's attached to, the carbon chain that it's attached to. So that's what we're going to use R as the whatever variable. It could be a long chain, it could be a short chain, it could be a ring. There's lots of things it could be. So that's hydroxyl. The second one is sometimes called a carbonyl, but I separated them for you guys into two different things, an aldehyde and a ketone. In general, carbonyl is a carbon with a double bond to an oxygen. But an aldehyde is when you have a carbon double bonded to an oxygen and it's on the end of some compound. So you have your attachment point here. That's your R, your attachment point. And then the actual aldehyde is this. Carbon double bonded to oxygen with another hydrogen attached to it. And a ketone is also a carbon double bonded to an oxygen, but a ketone is where it happens in the middle of a compound. So you have an attachment to some kind of carbon compound on the left and an attachment to some kind of carbon compound on the right, and then somewhere in the middle you have the carbon double bonded to the oxygen. So that's a ketone. The general word, I don't think I even included the word carbonyl in your notes, but the general word carbonyl refers to the carbon being double bonded to the oxygen. So the fact that you have this is a carbonyl. But ketone is where it's in the center of something, and then aldehyde is where it's on the end. So any questions about aldehyde versus ketone? And again, all you're responsible for, I actually made a Quizlet. It'll be available on Monday, or starting after Monday, matching the diagrams to the names so that you can practice it. All right, so the next one is called a carboxyl group. So carboxyl group, the way that it looks in a drawing is like this. It's a carbon double bonded to an oxygen and then an OH. Another way you could see it, if they weren't going to sketch it out, is they could just write COOH. So this is, you could draw both. This is the structural formula. This one, this is actually what it would look like, that it's a double bond to an oxygen and then a hydroxyl. And this one is not showing how it's spread out, but it's just sort of the formula. So those are two separate ways that we can write the carboxyl group. Yes? Yeah, this one. This one. Oh, the ketone.
Yeah, the ketone. So what is that different from the aldehyde?
The aldehyde is on the end. So the aldehyde, you have some carbon chain, and then you have the carbon double bonded to the oxygen, and then just a hydrogen here.
Okay. So
And this one, you have some kind of a carbon chain on the left, and another carbon chain on the right, and just the carbon double bonded to the oxygen in the middle. So it's not, this one's on the end of a compound, and this one's somewhere in the middle of a compound. And then this one has an oxygen, because this looks a lot like this, the aldehyde, but this one has an oxygen here. And the carboxyl, I believe, you have in your notes that it's also called carboxylic acid. Do you have that there for the carboxyl group? because it is, also called carboxylic acid group. I thought I had listed both names. So if I didn't, that is the other name for it, a carboxylic acid group. Because this hydrogen sometimes comes off. And that's the definition of acids. Acids are things that can give up hydrogens, or at least that's the definition we were working with, and that hydrogen sometimes comes off. All right, the next one is an amino group. The amino group looks like this. Here's the attachment point, the little R, and it's nitrogen connected to two H's. Or if they were going to shorten it down, they could just write that, just write NH2, because that's technically what it is. But structurally, it would look like this. N, a nitrogen, a hydrogen, and another hydrogen, or shorthand version, just NH2. And I'm going to erase the aldehyde and ketone stuff up here so we can make some space for the next ones. Alright, get rid of the carboxylic acid. Alright, so the next one, the sulfhydryl group or a thiol. Here's your attachment point, and it's literally a sulfur and a hydrogen. So that's a thiol. All right, phosphate you might recognize from chemistry. So a phosphate, this is our connection point, and it's a phosphorus surrounded by oxygens. Two of the oxygens do have a negative charge. So in this case, this actually is a charge here and here, and this one's double bonded. A shorter way of writing it, if they were not going to draw it out, is they could just write that. They could just write PO4. But the way that it physically looks is think of the phosphorus completely surrounded with oxygens all the way around it. Yes? This is just a shorthand way of writing this. So rather than actually showing what it looks like, where this, in other words, this is the same thing. It's just that this one is showing that it's double bonded to this oxygen and then single bonded to the other oxygen. And in this one, it's just like condensed. Yes. Carboxyl, or it's also called carboxylic acid. That's just two names for the same group. All right, so this was sulfhydryl or thiol. This was phosphate. I know this is getting messy. Methyl group is is basically, here's your connection point, and it's literally CH3. Or I'll draw it out, like a little space. It could also be drawn this way: carbon, hydrogen, hydrogen, hydrogen. So you see CH3, CHHH. That would be structurally how it would be drawn. This is the condensed way. All right. All right, last one is the ester. And I'm like totally out of space here. I'm gonna get rid of the carboxyl here to do the ester. Actually, I'll get rid of the amino too. Okay. So the ester group looks like, here's your connection point, and it's going to look like this: a carbon double bonded to an oxygen, another oxygen, and then it's connected to something else. So this whole thing is the ester. It's a carbon double bonded to oxygen, another oxygen, and then it's connected to some kind of a chain on both sides of that. Let me just... Alright, so the organic compounds we're going to talk about next week: carbohydrates, proteins, lipids, and nucleic acids. They're all made of building blocks. The building blocks are called monomers. And then when you put them together in chains, the chains are called polymers. So for example, proteins, the monomers of proteins are amino acids. You make a chain of amino acids, that's a protein. Carbohydrates like starch are made of monosaccharides. So the monomers, monosaccharides, the polymer is starch, cellulose. They're called polysaccharides. The monomers of lipids are fatty acids. The monomers of DNA are called nucleotides. But all organic compounds are made out of these building blocks called monomers, and they get put together. The way that they're put together is something called a condensation reaction or a dehydration synthesis. When you think about being dehydrated, what does it usually mean? What are you missing? What did you lose? Water. So in a condensation reaction or a dehydration synthesis reaction, that's exactly what happens. You start with two things. One of them has a hydroxyl on it, and the other one has a hydrogen on it somewhere. The hydrogen and the hydroxyl get together, react, and form water, and water would leave. And then when water leaves, the two original monomers get linked together. In other words, it sort of creates the space for them to bond. This is what your body is doing all the time. Anytime you're building stuff, when you're building proteins, hormones, enzymes, when you're building fingernails, when you're making hair, when you're storing fat, when you're building complex carbohydrates, that's what's happening. You're doing condensation reactions. Now, on the flip side of that, sometimes you need to break those things down. For example, you need energy, so you break down the stored fat. Or you digest your food. You eat a big meal, you're going to break down all of that into its basic building blocks. So that is called a hydrolysis reaction. A hydrolysis reaction is the opposite, where you take something complex and you split it with water. Well, I mean, it's present, but it was lost in the chemical reaction. It was released. So you hooked the two things together, and so it's called a dehydration synthesis because you synthesized a complex molecule, but it got dehydrated. It lost the water, so the water was lost in the process. But you're right; it's still floating in water. Yeah, and by the way, both of these reactions that are happening in your body. It's not just that you just add water to something and it breaks apart. Obviously, if I just squirt water on a piece of steak, it doesn't break into amino acids. There's enzymes and other things involved in these reactions. But this is the bottom line. Here's a diagram in 3 dimensions, or a little more. So this is a polymer, and here's a monomer. And notice here's a hydrogen, a hydroxide. An enzyme would help with this. The water is lost. It produces water, released. And now these are now hooked together. So this could be building like I said, a complex carbohydrate or a protein. Here's another one. This is actually just the opposite. So this is splitting it. Water comes in, and you get back where you started. This is the one that's in your notes. These are actually monosaccharides. And what will happen in this case is the OH from one, the hydroxide, and the H from another will hook together and make water. Here's the water. And the oxygen that's left behind will actually be the linkage. And these will actually hook together. This would be a disaccharide in a long chain. This would be a polysaccharide. And again, when you're doing things like digesting, you're breaking it up. When you're building, like for storage or for whatever reason, you're going the other direction. So your body can do both of these. Your body can build them. Your body can break them down. All right. So the two green things that look like jelly beans, these are your monomers. So watch what happens now. Here comes an enzyme. This is helping. Water is released, and they're hooked together. And now another water is released, and they're hooked together. And now you've built a polymer. Now here's the opposite. A different enzyme comes in, a drop of water is added, and it gets hooked back to them, and now they're split apart. So that's just to give you sort of a